We'll see how that works. Okay, so this is a tree, which represents plants. All right. And a plant, like any organism, has to be able to meet its basic needs. Well, what does a plant need to survive?
Water.
Water, sunlight, carbon dioxide, O2. Right? Needs a number of different things. And so the plant, like we are, is designed to supply the materials it needs and to get rid of the materials it doesn't need. Yeah? Plants like ourselves are multicellular, which means each cell cannot supply its own individual needs. They have to work together in coordination and help each other meet each other's needs, just like your own body. Yay? As a multicellular organism, we solve that problem. As a human, how do we make sure everybody gets what it needs, primarily? We have what type of system? How do we make sure every cell gets what it needs? Through what system? What's the delivery system?
The veins.
Which are part of what system? The circulatory system. Holy cow. I think we better retest for getting in here. It's the circulatory system. Well, plants also have a circulatory system, but very different for getting the materials we need in and out of the plant. So one of the primary things plants take in, of course, as you see in the upper right, is light. Why in the world does a plant take in light? Photosynthesis. All right. What part of the plant is designed to capture the light? The leaves. Particularly what part of the leaf? No stomatas. Chloroplasts. All right. The chloroplast is designed to trap the sunlight and bring it in. Why do we need sunlight? What are we going to do with it? We don't make energy in photosynthesis. We transform energy from light energy into chemical energy in the form of sugar. Well, what else do I need to make sugar? Water and carbon dioxide. How does the carbon dioxide get into the plant? It doesn't have lungs. It doesn't have a nose. It's got, what's stomata? What's a stomata? Opening in a leaf. Where are they usually located? Why on the underneath? If they're on top of the leaf, what's the problem? They're exposed to light. They also get in the way of capturing light. They're on the underside of the leaf. They're little openings that have the ability to open and close due to something called guard cells. Yeah? And so they allow carbon dioxide in. And what comes out of those? Oxygen and water. It's an exit route for water and oxygen. It's an entrance route for carbon dioxide. That's always a tricky balancing act for a plant. If my stomata are open, good news is I'm getting carbon dioxide to make sugars. The bad news is I'm losing water and dehydrating. So it's constantly a battle between water loss and carbon dioxide intake between the stomata. How does water get into the plant? Primarily through the roots. Also, that's the primary way that the nutrients get into the plant. What is the one product up here that does not go in or out of the plant? Sugar. Where does the sugar come from then? So where did it come from? How did the sugar get there? And who carried out photosynthesis? The plant. The plant made the sugar. You don't absorb sugar. You don't release sugar. You make sugar. You make food. The other stuff I got to either bring in or take out. Yeah? And then once I make the sugar, I got to send it where it's needed. And that changes depending on the time of year. Do I want to put it into the roots? Do I want to put it into fruit? Do I want to put it into leaves? Where do I need to put that energy? That varies depending on what's going on. So there are two main transport systems. You can see blue arrows and green arrows. Blue arrows is describing the transport of what? Water. And water is moved through a process called transpiration. So that's one of the things we're going to highlight today is transpiration. A little bit more detail than we've done so far. The green arrows represent the movement of what? Sugar. The green arrows represent the movement of sugar. Anybody recall what that's called? The transport mechanism. Water's transpiration, sugar is... What kind of notes did you take last night? Something called translocation. Translocation is the movement of sugar. So we're going to feature these two processes. We can see that the blue arrows all move up in one direction. It's a one-directional flow system. We're going to see it's also based on what we call negative pressure. It's a negative pressure one-way flow system, which really is completely controlled by what key factor? Cohesion is involved, capillary action is involved, but what drives the process? We spent 2 days on it. The last 2 days. Water potential drives the whole bloody thing. Okay? And she wasn't even here. So water potential is what drives the negative pressure system, we'll see, to drive transpiration of water. Oh my goodness, things might connect for a moment. You're not supposed to do that in school. You're supposed to do it, get it done, and forget about it for the rest of the year. Wrong. You mean I got to remember something I did 3 weeks ago? Uh-huh. And later on, you mean I got to remember something I did 3 months ago? Uh-huh. Yes, sirree. Sugar is called translocation. What do you notice about the green arrows? It's not. It can move in multiple directions based on where the sugar needs to go. The last couple of months, where was sugar moving? Down to where? Roots. Why? Beautiful. In the spring, where's the sugar going to move? Why? Provide the energy to leaf out. Midsummer, the sugars will move where? To the fruits and the flowers to produce the enticing treat for the organisms to come and disperse their seeds. Yeah? So that's sort of a general overview of what plants need to do. Like you and I, they've got to respire. They've got to move gases in and out. I'm hoping by now we realize that plants do both photosynthesis and cell respiration. Yeah. So they also not only release oxygen, they use oxygen. But they don't have lungs and a nasal nose and alveoli and trachea. They have stomata. Plants need water. They don't drink it. They don't walk over to the pond, bend over, and slurp up the water. But they have roots that have access to water to bring that water in, and so forth and so on. Questions on the overall diagram? So we're going to focus on what two processes here? Transpiration and translocation. So let's start at the roots. This is a picture of the roots. And it's not cooperating. Where'd my record button go? Yes, sir. For 1 second, yes. Only because you went to Michigan State. Oh, turkeys. So anyway, here's a picture. Sorry, I'm acting confused. What are you doing to me? Okay, we're back after that short intermission. Up here is an enlargement of a root. And here's the root hair, and here's the central part of the root. With me? Everything ultimately has to go to the central core of the root, or what we call the stele. Right? That's this region here that I'm going to circle in red. That is made up of the two primary vessels that make up plants. In humans, we have vessels, but we call those arteries, veins, and capillaries. In plants, what are the two primary vessels? Xylem and there's no way you read anything. Xylem and phloem. Xylem and phloem, the two basic tubes that run through a plant. Xylem is responsible for carrying what? Water and some minerals. Phloem is responsible for carrying sugar. So xylem and phloem are the two major tubes that are there. They're at the central region of the root. They're in what's called the stele. Surrounding them, which I'm going to highlight in blue here, is this what's called endomembrane or endodermis layer. This endodermis layer is a barrier layer. It is like the crossing guard at the Blue Water Bridge. Its job is to do the inspection before it decides to allow something through or not to allow it through. Okay? So it makes sure that everything that's going to the xylem and phloem, which means it's now got access to the rest of the plant, has been properly screened. And it's also preventing anything from the plant escaping that you've already gathered. Because again, a lot of times these roots are in very hypotonic environments, which means you would lose water and nutrients to the environment if this endodermis wasn't there to protect the loss. Going anywhere? Part of this endodermis is this little thread that's running through it called the Kasparian Strip. Whoops. That Kasparian Strip is a highly waxy structure, which means it's primarily composed of what? Lipids, which would make it nonpolar and hydrophobic. Right? So that forms a really nice barrier to allowing things into the stele or out of the stele. It's just as important that it keep stuff in as control what goes in. Yeah? There's basically two routes. There's the symplastic route, which if you're watching this at home probably got cut off. And there is the apoplastic route. They both work. They both are fine. It's just when you're going to do the inspection. Do you want to do the inspection up front or do you want to do the inspection at the end? Do you want to talk to the American Border Patrol or do you want to talk to the Canadian Border Patrol? Either way, we're going to get you. Got me? So, in symplastic route, the symplastic route, we're going to pass through this thick layer, which represents what? The cell wall, since it's a root of a plant. Right after the cell wall, we're going to encounter what? A plasma membrane, which would be selectively permeable. Therefore, we're doing the inspection up front. And then it enters the cytoplasm. If you follow my blue marker here, following the blue line. I went through the cell wall, the plasma membrane, right into the cytoplasm. Now that I'm in the cytoplasm, I can easily travel from cell to cell through these openings. What are these little joints between the cells called again if it's a plant cell? Plasmodesmata. Which might explain why almost everybody got the question wrong about gap junctions and plasmodesmata, even though you're supposed to make a chart comparing them. Nobody's got anything to say on that. Plasmodesmata. What's the advantage to having these gaps, these junctions? Bingo! You don't have to keep crossing the membrane. Crossing membranes take time. Look at this guy. If he wants to get all the way into the xylem, he's got to cross the cell wall and the membrane. Cell wall membrane, 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 cell wall membrane. Holy Toledo. That's going to take me a long time. It's like trying to go over to Canada and you got to stop at 15 different toll booths just to get across the bridge. You had to get out of your freaking car and jump off. Yeah? So by going through these Plasmodesmata, we save time. And in fact, when we get to the endodermis and the Kasparian Strip, there's these Plasmodesmata that let us go all the way through. We bypass the endodermis because we've already been checked. We've already been processed. We already know that we're going with the gradient. We've gone through all that water potential, osmosis, all that stuff. Yay? Another way you can go through is the apoplastic route, which is represented by the pink line. You're going to go into the cell wall, but you're going to travel through the matrix in the cell wall, which allows you not to have to, again, deal with membranes and cytoplasm. But what haven't you done so far? You have not crossed a selectively permeable membrane. So when you get all the way to the endodermis, you've not been inspected. You cannot cross the Kasparian Strip. We're going to force you to cross the membrane at the endodermis, where now you're going to get checked out. And if you check out, you'll be allowed to cross through the Kasparian Strip, through the endodermis, and into the steel. Yay? So 2 different ways we can move materials. There are slight advantages and disadvantages to each. All right? In both cases, though, the key is you have to eventually cross a membrane so that we can keep things in balance. And by the time anything gets to the steel, the xylem and the phloem, we know it is something that we need. And then it can be transported. Going anywhere? Yes, ma'am.
In our book, it showed a transmembrane route. Is that what
Transmembrane is when you do sort of a combination. So I start out apoplastic, and then I leak into this cell, and then I take on a symplastic route. Okay. But at some point, again, you have to cross the membrane to do that. Let's be crossing the membrane of the plasma membrane of that particular cell versus the endodermis. It's just an enlargement. All right. Here's that symplastic route where you're using the plasmodesmata going directly in. Here's the apoplastic route, which is sometimes good, particularly for bigger molecules. And then they only go through the process at the end when they try to go through the endodermis. And that's how we screen minerals and screen water before they get into the xylem. Yeah? Once we're in the xylem, now it's the transpiration, baby. Okay? And it's all about creating negative pressure. If you look at this strip right here, it'll be part of your key tonight. In the soil, our water potential is at negative 0.3. In the atmosphere, it's at negative 100. That's a huge difference. Way more positive here, closer to 0 here, way more negative here. This is why we say it's a negative pressure system, because water is always going to move from high potential to low potential. That creates a natural, almost suction to pull the water up, which then also takes advantage of those properties of adhesion and cohesion. Yay? All right. I'm going to obviously run out of time before I can finish this brilliant discussion. You will